In metabolic processes, a lot of what's happening is that electrons are being transferred from one molecule to another. This sometimes allows energy to be transferred from one molecule to another in very small bits. When a molecule has an electron, it's reduced. When a molecule gives up its electron, it's oxidized. Gets the electron, it's reduced. It gives up the electron, it's oxidized. Some molecules are better at accepting electrons than others. Oxygen is the very best electron acceptor. It's very electronegative. When oxygen gets near anything, it just takes any available electron. I've been oxidized! That's why we call it oxidized. When a molecule loses its electron, it's as though oxygen has taken it away, so it's oxidized. I can become reduced again when I accept another electron from somewhere else. The first step of cellular respiration is glycolysis. In glycolysis, the name really tells you exactly what it's going to do. It's got two parts, glyco and lysis. Glyco means sugar, and lysis means split. So the process of glycolysis just splits sugar into two. So we have sugar, which is a 6-carbon molecule, and glycolysis just splits it right in half. Okay? So now we have 2 molecules which each have 3 carbons, and these are called pyruvate. So the results of glycolysis are 2 pyruvate, 2 ATP, and 2 electron carriers. And that's what I want you to know. Glycolysis creates a little bit of ATP and a little bit of electron carriers, and all life forms, every single thing, heterotrophs, autotrophs, eukaryotes, prokaryotes, they all perform glycolysis. The next step is called pyruvate oxidation, and that's very simply where the pyruvate, which are these 3 molecules, get broken up just a little bit more. So basically what happens is that these pyruvates get one of their carbons removed, each one. These carbons, when they get removed, they become CO2. Remember, oxidation happens when oxygen absorbs electrons. These carbons have become oxidized. They have been connected to oxygen. So the result here is 2 carbon dioxides, and we also get 2 electron carriers. There's no ATP produced in pyruvate oxidation. The next step of cellular respiration is the Krebs cycle. And in the Krebs cycle, acetyl-CoA, 2 carbon molecules, enter a cyclic metabolic pathway. So when they go through this pathway, they're changed multiple times, and these carbons are stripped off one at a time to make more carbon dioxide. But in the process, as they go through this cycle, we get 1 ATP and 4 electron carriers. Now, since we have 2 of these acetyl-CoA, 2 carbon molecules, it has to go through the Krebs cycle twice in order to break down the entire glucose molecule. So since we go through twice, we end up with 4 CO2, 2 ATP, and 8 electron carriers. Okay, so far we've only created 4 ATP molecules, and the whole point of cellular respiration is to create ATP. But we do have all of these electron carriers that have been produced. These electron carriers help in something called oxidative phosphorylation. It's a big complicated word, but it just means that we're able to make a lot more ATP using a pressure gradient. So let me see if I can show you sort of how that works. All of these electron carriers get shuttled to the inner membrane of the mitochondria. They're already inside the mitochondria, so they're right there ready to go. These electron carriers pass their electrons onto an electron acceptor in the membrane of the mitochondria. So the electron gets passed to a molecule that's just a little bit more electronegative than NADH or FADH2. But then this molecule is next to another molecule that's a little bit more electronegative. So it accepts the next electron. And then this one is even more electronegative, so it accepts the electron. So the electron is being passed from enzyme to enzyme in the membrane of the mitochondria. The final electron acceptor is, you guessed it, oxygen. When oxygen accepts that electron, it combines with hydrogen to make water. Okay? Now, actually, all of these molecules are combining with hydrogen when they accept the electron. So the hydrogens are going into that enzyme, but then when the electron is passed, the hydrogen is pushed into this little space between the two membranes of the mitochondria. So hydrogen is pumped out of the inside of the mitochondria and put into this space between the two membranes. And so what happens is there's a lot more hydrogen in this narrow space between the two membranes than there is out here. So it's sort of like a pressure gradient. So since there's so much hydrogen in this area, the only place for it to escape is through this very special molecule called ATP synthase. The hydrogen, there's a special channel just for the hydrogen. So it goes into this molecule, and when it does, it actually spins, just like a pinwheel. So this molecule rotates, the ATP synthase rotates, and when it does that, it creates enough energy to turn ADP into ATP, which is what we want. So the electron carriers passing their electrons along the electron transport chain pumps hydrogens in between the membranes of the mitochondria, which creates a pressure gradient, which allows this ATP synthase molecule to turn and connect phosphorus onto ADP, making ATP. So oxidative phosphorylation creates a lot of ATP. We either get 32 or 34 molecules of ATP, and plus we get a lot of electron acceptors. And those electron acceptors can go back to glycolysis, the Krebs cycle, and so the whole process can keep going.